All right, class, let's get started with our lecture. All right, just like we were talking about in lab, we're talking about DNA, replication, binary fission, and mitosis, right? So cells need to have the ability to be able to replicate, all right? Be able to replicate. It's part reproduction, it's part of growth, it's part of repair, it's part of development. And so it's something that has to happen for prokaryotes and eukaryotes to be able to stay alive, right? So all cells divide, right? Obviously, if cells didn't reproduce, they would know, once they died, they would, that whatever that is would no longer exist. But because we're able to reproduce, not only in the instance of repair or rebuilding, but also when reproducing, like reproducing another, the next generation. All right, so unicellular organisms, uni meaning one, single-celled organisms, bacteria, for example, viruses, can divide in order to reproduce. Cell division produces a continuous supply of replacement cells and multicellular organisms, all right? So as human beings are multicellular organisms, right, Ms. Kadira? Multicellular organisms. And so we have to be able to reproduce. So you think of it, you cut yourself, like you had an injury today in class, cut yourself, you get injured, you put a Band-Aid on it. What happens? That cut doesn't just stay there forever. It eventually heals. New cells are formed, right? From other cells reproducing forms new cells, right? So that has to that happens for us to be able to do things like to heal or to grow or, all in many different ways. But those are just some common examples. Eukaryotic cells divide by mitosis, all right? So a fertilized egg or a zygote, right? So you had a question? Oh, I thought your hand was up. Mom and dad come together, sperm meets egg, forms what is called a zygote. All right, so we all start off as a single-celled organism with a full set of DNA, chromosomes, full set of chromosomes in our DNA, right? But obviously, by the time you're born, that one cell that you started off with, that fertilized egg, has multiplied many, many, many times, right? So mitosis, so the fertilization, the sperm fertilizes the egg. You have what is called the zygote. We all started off as a zygote. And then we go through the process of mitosis. So split cells splitting. And in this case, the mitosis happens, but also we have the process known as differentiation, which we'll talk about later. But some cells go to be the eye cell, some cells go to be the arm cell, some cells go to be the muscles, some cells go to be bone, and cells just keep splitting and forming new parts of the baby, and then eventually the baby's born. Okay? You also have the process known as meiosis. So each mature individual produces sex cells, right? So when so your sperm in men, eggs in women, each ones have different numbers of, have half the number of your DNA, your genetic material. And so meiosis occurs only during reproduction. So the idea of making sperm or making eggs only occurs during reproduction. So if half of my DNA, half of my wife's DNA went into each one of our children, and it's not necessarily the exact same half, where they would be all identical, but they're not. All right, so it's a mixture of half of my DNA, a mixture of half of her DNA, and then that came together to form, that was the sperm, that was the egg, and they came together to form what is eventually known as the zygote. But you have the making of your sperm and eggs is meiosis. That's going to be making of the sperm, which is going to be half of the man's DNA, or the making of the egg, which is half of the female's DNA, right? You are half of your genetic makeup is from your father, half of your genetic makeup is from your mother, whether you like it or not. That's the way we are designed as human beings.
If only we could pick what to get from them.
What if we could pick what to get from them?
While we're in our mom, while we're in the belly, we just get a screening of traits and
You
all
mean
this.
character creation?
You mean like I wanted like my mom's eyes, my dad's lips? You talking about something like that?
All around.
All around characteristics.
He's talking about when you open a video game.
Characteristics.
Oh yeah. Oh, well, like when you make a what do they call it?
Cons, anger issues, all that.
Anger.
Ears, I want this ear.
So meiosis is half the DNA. We just talked about that, right? And then mitosis, which we talked about in lab today, mitotic cell division allows for an organism to grow, which is happening when you're in the mother's womb, and develop. Also repair. Talked about that when you have like an injury, your cells must grow to be able to repair and regenerate lost body parts. Some organisms reproduce asexually without another organism. But human beings reproduce sexually. That means we have a male and a female come together to make the next generation. Okay? Mitosis produces body cells. Okay, so this is showing you the process here. We start off as day one, just one egg that has been fertilized by a sperm. We eventually start to divide. You see it's a whole bunch more cells here. Then you got all that group of cells there. And then you start to you keep going. You eventually start to form structure. And we see after many rounds of mitosis, multiple cells splitting, forming new cells, copying the DNA, you start to see this shape of a baby in a matter of less than 2 months. Cell death is also a part of life, right? The cells that you have right now in your body are not the same cells you were born with, right? Some cells have died. Like I said, red blood cells only live about 100 to 120 days. So those cells the original blood that was pumping through your body, all that blood is gone. You've replaced those blood cells over and over again. Your skin, for instance, the surface of your skin is actually dead skin cells, right? So your skin is constantly making new cells at its deepest layer, and then those cells slowly move up as the cells on the top. Those of you who go and get manicures and pedicures, especially pedicures, when they use that paddle thing and they rub the bottom of your foot, they're removing dead skin cells. That's what they're removing, cells that are on the surface that are now dead cells, and they're removing those and makes your skin look nice and shiny, but all they're doing is removing the dead skin cells, right? And so other cells are constantly growing under those cells, and it's slowly moving to the top. So as cells on the top layer fall off, which is natural, then other cells are slowly moving up. And so there's a process of mitosis that's happening as skin cells are dividing over and over again. But it's normal for cells to die. Most cells in your body do not live forever. Well, nothing in your body lives forever, but most of the cells in your body are going to be replaced at some point in time, right? So we have what is called apoptosis, cell death, process that can carve out distinctive structures during development, and certain cells may die and be replaced. I'm going to do the questions right now. DNA replication is before cell division, all right? So remember we talked about interphase, nothing is going on, and then we have metaphase. We see cell division, a DNA replication before we have cell division. So for each of the daughter cells from this division to have identical DNA, so for one cell to divide and make two cells with the same amount of DNA, the cell must first replicate its genome, all of the cell's genetic material, right? So every cell in your body, you need to understand this, every cell in your body has DNA. Every cell in your body has DNA, all right? Or had DNA at one point, right? Some cells, once they're about to die, they no longer have DNA, they kick the DNA out. But every cell in your body that has a nucleus has DNA. The DNA in all of the cells in your body is the same, all right? That's why if you go and commit a crime, not to say that I would recommend anybody committing a crime ever, but if you go and commit a crime and they have a piece of your hair, or they can get a piece of dead skin that was left from your fingertips, or any skin from your body, or any hair from any part of your body, and we take that to the lab and we do a DNA test, whether it's from your skin, whether it's from your hair, whether a hair on your head or a hair on a piece of your body or something else, if we do a DNA test, it's all the same DNA. It'll all be you, all right? And that DNA is unique to you. All right, there's no one exactly like you, unless you have an identical twin, then we have maybe a little bit of a problem. But for the most part, there's no one exactly like you. All right, and so whenever a cell in your body has to duplicate, goes through the process of mitosis, making a new cell, one of the first steps we have to do is copy the DNA, because we need that new DNA in that new cell. Okay. So the cell must first replicate its genome, all of the cell's genetic material. And so DNA is deoxyribonucleic acid, and it's gonna house all of our genetic information. This is the code for you. All right, it tells your eye color, your ears, your the size of your brain, how tall you are. All of those things are coded for in your DNA, right? Your DNA. Now, we don't understand every aspect of DNA. Like we've mapped the DNA. If you've ever heard of the Human Genome Project, we've mapped the DNA. We've tried to figure out which parts of the DNA code for which things in our body, we know certain things. We know, like, for instance, male, female. We know, for instance, certain diseases. We can look in the DNA and see how someone would be susceptible to certain diseases because they have this gene or that gene, or don't have this gene or don't have that gene, for instance. All right, so your DNA is your genetic code. Everybody's probably heard of DNA. It's in the news with the 23andMe and the different. What do you call it? What do they call it? Ancestry.com, all of those. And what are they doing? They're taking, we know that your DNA is original, but we also know that people who are related, remember, your DNA came from half your mom, half your dad. Their DNA came from half their mom, half their dad, and back through history, right? So if I have your DNA, and if I have, a million other people's DNA, I can kind of see trends and say, okay, well, these are somewhat similar, these two people or these groups of people. And I can say, oh, you're probably from somewhere, in East Africa, for instance, as an example, because your DNA is similar to people from this region, right? Because we're all related. Our DNA is all similar to somebody, our parents and our parents' parents and our parents' parents' parents, and all go back through the line. All right. So that's a whole. So once we mapped out DNA, and now, people are taking swabs of their saliva, of their cheek, inside of their cheek, which is the cells on the inside of their cheek. They're sending it in, DNA sample. people are getting their DNA mapped out, and then the more people you have, the more you can compare and see the similarities and say, oh, you might be related to this person, and you might be related to that person, might be related to that person, and you all may be related to people from this part of the world, as an example. Okay. Anybody done that? Anybody done 23andMe, Ancestry.com? You've done? Okay. Oh, I thought you were going to give us those. You did it and you found out something. Oh, okay. Yeah. You said you were, hold on. You said you were going to do it? Okay. Okay. Anybody did it and found out? Okay. Did you find out anything interesting or where you're from or?
I found out I was like Polish.
Okay.
That's true.
Yeah. It is.
Better as the years go on.
Well, the more that people put their DNA into it, the more information they have, the more data they have, the more accurate it could be. All they're doing is taking your DNA.
Oh, like your blood or something?
Well, yeah.
No, you can get it from the inside of your cheek. You can just swab your cheek. You don't have to give them blood.
And what do you find out?
You just find out. You'll find out like they can tell you like if there's people who have sent in their DNA that you are maybe closely related to, and they can also tell you like where your ancestry is from. Like, is your family originally from like Poland, for instance, or from East Africa, or from somewhere else?
So they're only taking your DNA. How can they tell you who are you related? Like I understand. Like
Because other people have submitted theirs. And that's what I'm saying. The more people submit theirs,
they combine them together.
right? And so like for instance, like if we were related, if you submitted yours like 5 years ago, and then I submit mine today, they'll be like, oh, you related to somebody who submitted theirs 5 years ago. We can, if y'all want to get in contact or whatever like that.
One more thing. So basically, I'm trying to understand what mitosis is. So mitosis is the separating of a cell, or is it like
It's the copying of a cell's DNA. Yeah. So the copying of your genetic information. The process by which that happens. All right? All right, so DNA replication builds 2 new DNA strands. So we copy the DNA. So we have 2 copies of the DNA. That's the whole point. And then the DNA helix is unwound. So enzymes called helicases, that's an enzyme, unwind the DNA. Single-stranded binding proteins hold the strands apart, and then we can copy the DNA. An enzyme called primase adds a short strand of RNA to each template strand. This is just the process. And then DNA polymerase is the enzyme that makes new DNA strands starting at the RNA primers. All right, so we split the DNA apart. DNA is normally 2 strands. We split it apart. We add a little piece of RNA to prime it. That's a little starting point. And then the DNA polymerase comes and copies the rest of each DNA. So from one DNA, we get 2 strands of DNA. The RNA primers provide a 3' end where the DNA polymerase adds the nucleotides. So adds the copied nucleotide. On one strand of DNA, the polymerase follows the helicase as it unzips the DNA. This is the leading strand. So leading strand as it unzips. And then you also have on the other strand of DNA, the other DNA polymerase adds nucleotides in the opposite direction from the helicase movement. And this is the lagging strand. So we have a leading and a lagging strand. So it's making 2 strands of DNA. And then an enzyme replaces the RNA primer with DNA. That RNA primer that was at the very beginning replaces that with DNA. And ligases form a covalent bond between the DNA segments. So make sure the DNA is nice and connected. So DNA replication has many points of origin, so you can start in different places. Simultaneously as multiple spots along the chromosome. Remember, the DNA is really long, right? So we're not necessarily just starting at one end and going all the way to the other end. We can start at multiple locations and start copying it. So multiple spots along the chromosomes. These spots are called origins of replication. So I can start in different places. Oh, I'm going to ask questions right now. All right. And prokaryotes, remember we have prokaryotes which have their DNA not bound in a membrane. Reproduction occurs by what is called binary fission, an asexual process that replicates DNA and distributes it to two daughter cells. So binary fission, remember their DNA is not membrane bound in the nucleus. It's kind of floating around. They divide it, split it, and then split the cell. Similar idea. Prokaryotes have a singular circular chromosome. Cell membrane, DNA, cell wall. As the cell prepares to divide, the DNA is replicated. DNA is replicated and attaches to the inside of the cell membrane. Remember, the cell membrane is a structure that sort of houses the cell. So DNA is replicated, attaches to the inside of the cell membrane, extends and grows between the two. The cell membrane extends and grows between the two DNA molecules, and then eventually partitioning off, forming two identical daughter cells. All right? Called binary fission when it's a prokaryote. In eukaryotes, like human beings and plants, it is called mitosis. Eukaryotes divide by mitosis. So eukaryotic cells do not divide by binary fission, so they divide by mitosis, which is similar but a little bit more complex. And that's what we did in lab today. Right? Replicated chromosomes condense before eukaryotic cell division. Right? So remember, we saw this sort of nothing's happening there. Just a big circle of DNA loosely packed. Right? That's going to be your what phase? Loosely packed DNA, just a circle. Interphase. All right. Loose in the nucleus, the DNA in the cell on the right has condensed into visible chromosomes. Right. That's when we start the process. Once the DNA condenses into visible chromosomes. DNA replication and protein synthesis occur when DNA is loosely packed. So starts off with DNA loosely packed. Cell division is occurring when DNA condenses into chromosomes. Right? That's on the left. That's DNA condensing into chromosomes. A little bit different. Naked DNA. So remember, DNA is these long chains, a whole bunch of information in your DNA. Your eyes, your ears, your mouth, your muscles, your bones, your organs, all those things, your brain, all that's in your DNA, coded for in your DNA. So it's a big, long list of all these codes, multiple chromosomes. So naked DNA wraps histone proteins. So this is, these are the strands of DNA. All that is your code for your DNA. It wraps around these proteins called histones. And the DNA wrapped around those histones is called a nucleosome. Nucleosomes cluster together into what is called chromatin. So DNA wrapped around different histones, clustered together in what is called chromatin. Scaffold proteins within chromatin keep it tightly packed together. And that's where we get, from chromatin is where we get chromosomes. So after DNA is replicated, discrete chromosomes form. Chromosomes. So highly folded chromosomes take up less space and are easier to move than unwound chromatin. Right? So you have this loosely packed chromatin, these DNA wound around these histones, and then they get packaged up together and make up what is called chromosomes. How many chromosomes do we have? 23 from mom and 23 from dad equals 46. Yes. Many different events occur in one round of cell division. Now, this should look familiar. We saw this today. All right. During interphase, the cell replicates its DNA and carries out many functions unrelated to cell division. In G1 phase, the cell grows and functions normally. All right, that's just normal cell growth and function. That's when, so every protein in your body is coded for in your DNA. And so if I need to make a new protein in my body, it goes to the DNA in my body to get the code for the amino acids for that protein. All right, so this protein synthesis can happen in G1 phase. G0 phase, the cell continues the function, but it does not replicate its DNA or divide. Most of your cells are currently in this phase. S phase, the cell replicates its entire genome, every chromosome. Now it is committed to divide. So DNA replication happens in S phase, right? During G2 phase, the cell produces proteins that are necessary for mitosis, right? And so generally you're in one of these, the cell is in one of those phases, okay? So cells leaving G2 phase, so you've replicated your DNA, and now you're about to start the process of, I've replicated my DNA, so now I have 2 copies of DNA in one cell, and now I can start the process of mitosis, which is basically cell division, taking those 2 copies of DNA and making 2 different cells, so each one has the exact same copy of DNA. All right, so cells leaving G2 phase enter mitosis, the division of the nucleus, which is followed by cytokinesis. Cyto means skeleton? No. Cyto.
I think about cytoplasm.
Cytoplasm. Where do we find cytoplasm? Inside what? The cell. Cyto means cell. Cell. Cytokinesis, forming a new cell. All right, so the division of the cell itself, cytokinesis. All right, so you have you've already copied the DNA. Mitosis, now you split the DNA to 2 ends, and now we break apart the cell into 2 different cells. And that is what gives us, that's the whole process of mitosis. Mitosis and cytokinesis, all right? This has to happen for us to be able to continue to live, right? We have to be able to form new cells, when they're cell repair, cell growth, development, all the things, okay? So mitosis is divided into 5 phases, summarized here. G2, late interphase, cell, we have complete DNA replication. And then early prophase, your chromosomes condense, right? And we can see those in prophase. Spindles form as centromeres move. So your condensed chromosomes, start to see them. Prometaphase, we didn't talk about this in lab, but nuclear envelope breaks up, spindle fibers attach. Your metaphase, they start to line up. Anaphase, they start to pull apart to each side. Telophase, now you have DNA on one side, DNA on the other side. And then cytokinesis is when you actually split the cell, right? In animal cells, the cleavage furrow first appears in telophase. That's telophase at the end of, basically at the end of mitosis. You have this cleavage furrow that appears. So you start to see an indentation here as the 2 cells are about to divide. It deepens as a ring of proteins beneath the cell membrane contracts, right? You can see cytokinesis in process here. You can see the cell wall. This is a plant cell. Okay. Compared to regular body cells, you may have, I'm sure everybody's heard of cancer, right? Why is cancer so problematic? Well, that's one reason. Right. So, compared to regular body cells, cancer cells have distinct features. They lose specialization, which means they don't, your nose cells, your taste buds, your ears, your arms, your bone, your skin cells, all those things, those are cells that are specialized, right? Your skin cells, protect you from the outside. They're waterproof. They can stretch. They can do certain things your skin cells can do. Your bone cells are very different from your skin cells. Your bone cells are nice and hard. They provide structure to your body. Your bone actually helps store calcium. So different things your bone cells can do. Cancer cells don't do anything beneficial to the body, right? They don't do anything. So they lose specialization, right? You hear of this like mass. They'll say like tumor or mass. Because it's just a glob of cells. It could be small, hopefully, or it could be big, unfortunately. But it's just a mass of cells that don't do anything. Don't do anything to help your body. We'll put it like that. But they can continue to divide, all right? Your body, you don't just get skin on top of skin on top of skin. Just like your skin doesn't just keep growing out for no reason, right? Your skin grows as necessary and replaces as old skin falls off, new skin is slowly being made, going through the process of mitosis. It's a continuous process. But your body makes what's needed, right? You don't just grow two noses for no reason, okay? It's not going to happen. Cancer cells, on the contrary, grow for no reason, right? They basically just continue to divide and grow, right? That's why it's a mass, it's a tumor, it's a lump, because it's just growing for no reason. It's just growing just because it can. And they can regenerate the ends of their chromosomes, all right? So that's why cancer is so dangerous, is because, and I'm sure everybody's probably known somebody with cancer, or you've talked to somebody or met somebody, or you've read about it, or you've seen it on television, but it can be very detrimental because these cells grow out of control. And I remember I was on a plane when I was in college, so when I was kind of you guys' age, I was on a plane, and I remember there was a guy sitting beside me, and he was, we were flying to Houston actually, and he was saying how, so this is like 25 years ago probably. And so he was saying how, at that time, the main thing was chemo and all that kind of stuff. And something he said that I thought was very interesting, because he was like, chemo kills cancer. And I was like, okay. He said, but the problem with chemo is it also kills other cells in your body. I said, he was like, so he's like, if we could develop a way, and they have developed some techniques to be able to try to pinpoint how to kill cancer cells. But if you know anybody going through chemo, it can be, it's detrimental to you're basically poisoning your body, but trying to poison the cancer cells the most. That's kind of the idea with chemo, which is not very good. But there are some other techniques in trying to remove the cancer cells or cut it out, or all kinds of things like that. They do try to, surgical techniques, medical techniques to try to stop the cancer. The other issue is if you ever heard of metastasizing, you know what that means? So the other problem with cancer cells is that, let's say you have cancer somewhere in your body, that cancer can spread. And that's huge. That's very problematic. You'll see, if you ever heard of, they have levels to cancer, like stage 1, stage 2, 3, and I think the worst is stage 4 cancer. That's when it has spread in other parts of your body. And so if cancer gets into your blood or gets into your bone, your cancer can spread to other parts of your body, and then it's like it can become very problematic, because now you can have cancer all over. And what do they don't want to to kill the cancer, they'd have to basically kill you. And so that's when it's very problematic. So try to limit yourself from things that would cause cancer, like smoking, drinking. You know, there are some things we can limit ourselves from. Now, some cancers are going to form no matter what, but we can try to reduce some of the factors. All right, cell cycle is under tight control. Obviously, we don't, just like we talked about with cancer, we don't want cells just growing for no reason or duplicating, going through the process of mitosis for no reason. It is something that needs to be controlled, right? You don't want to just grow another head, for instance, for no reason in your body. We don't do that because our body has controls on, and that's why our body doesn't do that, all right? So you have chemical checkpoints that regulate the cycle. Checkpoints ensure that the DNA is replicated. The DNA is not damaged. Chromosomes lined up and separate properly, right? So you have different checkpoints along this process to make sure that the cell cycle is under control. Cancer cells are not under control. So cancer cells, your body has lost control of cellular division. So cells continue to divide even when they don't need to, right? That's the problem with cancer. You may think growing is great, but yes, uncontrolled growth and growing without a purpose. Remember, these cancer cells are non-specialized, so they're just cells that are just blobs of cells growing without a purpose. They still require energy. They're still alive. They're not dead. They're growing, but they're not dead. So they're still sucking energy from your body, and just cells growing and growing and growing is not good for you. Cancer cells divide out of control, can form a mass called a tumor. Tumors are either benign or malignant. So benign, they're contained to one spot, and sometimes they're removed when they're benign, or malignant. They can be malignant. Malignant cancer cell can spread to other parts of the body. Cancer arises when genes are mutated. So remember, we have DNA. Sections of your DNA code for different things. Those are called, different sections of your DNA are called genes, and mutations can alter the function of key cell cycle control genes, and cancer can arise. So cancer arises from overactive proto-oncogenes. If you ever heard of cancer, the study of cancer is oncology, right? That's from this term right here, proto-oncogenes, or from underactive tumor suppressor genes, right? Can lead to when your DNA basically mutates or something malfunctions with your DNA, it can lead to these proto-oncogenes, which can lead to accelerating the cell cycle, which can lead to cancer cells, right? So these different types of mutations in your DNA, a changing of your DNA. Remember, your DNA is supposed to be the same in every cell, but if a cell goes under a mutation, it can lead to forming cancer cells. Cancer risk, this is something that we can find out through 23andMe and other different DNA sites. Cancer risk can be varied by ethnicity, and so that's why you do a map of your DNA, we can see, oh, you're predisposed to some type of cancer, or more likely to get, even if it's not cancer, more likely to get this disease or that disease, which can help you, make some decisions in the future, right? So gene mutations, some can be inherited, making them more common in particular ethnic groups, right? This example, BRCA1, increases the risk of breast cancer. So you can get your DNA mapped out and you can see if you're at higher risk for something like breast cancer based on your DNA. Reducing DNA damage reduces cancer risk. All right, so inherited mutations and factors in the environment that damage the DNA. Factors in the environment that damage the DNA, like smoking, is one of the factors, can increase cancer risk. Mutations are caused in many ways, right? So eat a healthy diet, avoid obesity, don't smoke, UV radiation can also cause, obviously, skin cancer. And certain viruses can also cause cancer, right? So just be careful. Different ways these certain things can lead to a higher risk for mutating your DNA and potentially having cancer. Surgery may remove tumors. You can surgically remove a mass, a tumor, but cells with DNA mutations may already be spreading through the body, right? So cells may have mutated, and when we go in to remove the tumor, it's not a guarantee that the doctor gets all of the cells that have mutated, right? There could be cells that are not in that tumor that have already mutated the DNA that are traveling through other parts of your body. So that's why they'll do, they'll go in, they'll do the surgery, but then you got to come back in like 6 weeks and we got to check again to make sure it's not like spread to somewhere else. Okay? Drugs such as chemotherapy, I already kind of mentioned those. Drugs such as chemotherapy and radiation can selectively kill cancer cells, which are physiologically different from normal cells. All right, we talked about apoptosis already, but it's a set of coordinated events that dismantle a cell from the inside out. Right, so cell death, apoptosis, cell death. A receptor on a cell receives a signal to die. Certain cells, proteins and enzymes begin to break down the components of the cell. Eliminates excess cells, carving out structures. So sometimes there's a reason for that cells need to die. And so weeds out aging or defective cells. So like, for instance, like your red blood cells, when they've been traveling through your body for 100 to 120 days, they get old. They're no longer as effective. Those cells we need to stop using those cells and make new ones, right? Goes through the process of apoptosis. Tumor cells mutate and divide quickly, often developing resistance to drugs. That's a little bit more on the cancer. One class of drugs does not target all cancer cells. Instead, drugs attack the blood vessels that supply the nutrients to the tumor. That's another way that we can attack the cancer cells. Remember, cancer cells are, like I said, they're still alive. And so one way to attack cancer cells is attacking the blood vessels that supply the cancer cells. They still need nutrients. They need oxygen. They need nutrients to be able to live. All right, tumors need blood vessels to grow, so we'll attack the blood vessels, reduce the tumor growth. That's one way we can do it. And that's the end. All right? Wasn't too bad, right? It's a long day, I know, but it's a good day. So we talked about mitosis and cell division and interphase, metaphase, prophase, anaphase, telophase, G2, G1, G0, S, growth, development, all the things. Cancer, uncontrolled growth. So I'll download it and put it online, and you can watch it again if you want to. Right? Have a good rest of your week. Oh, it's only 44 minutes. That's not bad at all.